Your body makes creatine. You can also eat creatine, and you can supplement it. These three sources are often treated as interchangeable, but they're not. And understanding how they differ explains some otherwise puzzling findings in the creatine research. Endogenous creatine I, the creatine your body synthesizes internally, is produced in the liver from arginine, glycine, and methionine at a rate of approximately 1 gram per day. This production is automatic, continuous, and not meaningfully influenced by dietary manipulation. You can't eat more glycine and expect your liver to produce more creatine. The enzymatic pathway is rate limited in ways that resist simple substrate manipulation. This endogenous creatine is released into circulation and taken up by tissues with creatine transporters, primarily skeletal muscle and the brain. It's chemically identical to the creatine you get from food or supplements. The molecule is the same regardless of origin. Exogenous creatine from diet comes primarily from animal muscle tissue. Beef, pork, fish, and poultry all contain meaningful creatine in raw form, approximately 2 to 5 grams per kilogram, varying by species and muscle type. Cooking degrades dietary creatine. Heat converts creatine to creatinine at a rate that depends on temperature and duration. Prolonged high heat cooking, braising, pressure cooking, grilling past well done, destroys a significant fraction of the creatine content of meat. Rare to medium cooking methods preserve more dietary creatine. This is one reason that the dietary creatine contribution for omnivores is lower than raw food composition tables would suggest. A person eating a large amount of well-cooked meat might be absorbing considerably less creatine than they'd expect based on raw weight data alone. The third source, supplemental creatine, bypasses the cooking problem entirely. Creatine monohydrate powder is chemically stable in dry form, and when dissolved in water and consumed promptly, it is absorbed effectively through the small intestine. Supplement creatine is the most reliable and precise way to raise muscle creatine stores because you know exactly how much creatine is reaching your gut in usable form. Food creatine involves uncertainty. Cooking method, temperature, duration, and individual variation in absorption all introduce variability. The total daily creatine from synthesis plus diet in it can be estimated but not precisely measured without laboratory tools. Supplement creatine can be measured exactly. For individuals who want to be confident they're reaching full saturation, supplementation removes the guesswork. For vegetarians, supplementation is not optional if creatine saturation is the goal. It's the only exogenous source available. For omnivores, the question is whether dietary creatine intake is reliable and high enough to contribute meaningfully to muscle stores. And for most people eating cooked meat, the answer is that it helps, but it's insufficient for full saturation. 3 to 5 grams per day of supplemental creatine, on top of whatever diet provides, is the reliable path to full saturation for virtually everyone. The source of the creatine doesn't matter once it's inside your muscle cell. What matters is whether enough of it got there. There's an additional consideration in the exogenous versus endogenous comparison that affects athletes specifically: the interaction between creatine sources and training adaptations. Regular intense training upregulates creatine transporter expression in skeletal muscle. Exercise is one of the stimuli that increases the number and activity of SLC6A8 transporters on muscle cell membranes. This training-induced upregulation means that athletes who train consistently have a higher capacity to take up creatine from the bloodstream into muscle cells than sedentary individuals. Supplemental creatine in a trained individual reaches muscle stores more efficiently and may saturate them more completely than the same dose in a sedentary person. This is part of why creatine research in athletic populations shows such consistent results. The population most likely to supplement creatine is also the population with the highest transporter capacity and the greatest performance sensitivity to phosphocreatine expansion. For sedentary individuals starting creatine, the transporter upregulation associated with beginning a training program compounds the benefit. You're loading more creatine into muscles whose capacity to absorb and use it is simultaneously increasing. The combination of starting supplementation and starting training is more effective at building muscle creatine stores than either alone. The reliability of supplemental creatine, precise dosing, no cooking variability, no dependence on dietary patterns, is one of its underrated practical advantages over dietary creatine sources.